Hello friends, we are discussing elongation step in prokaryotic translation. In the first part of this video, we discussed about its definition, then we discussed about elongation factors, we discussed about three types of elongation factor, EFTU, elongation factor thermo-unstable, EFTS, elongation factor thermostable, and EFG, that is also known as translocase. Then we discussed about three sites of ribosome, P site, A site and E site. We saw the overview of elongation with the help of flowchart. Then we saw the diagrammatic explanation of the translocation, elongation step. In this part, we will discuss about elongation process in detail. We will discuss about the three steps of the elongation. Then we will discuss about EFTU, EFTS exchange cycle. Then we will also see the peptide bond formation, and at last we will discuss about some facts related to elongation. So stay tuned. Now let us see this elongation process in detail. This elongation process includes three steps. First is aminoacyl transfer RNA binding or decoding. Second is peptide bond formation or transpeptidation, and third is translocation. Now let us see these steps one by one. So first is aminoacyl transfer RNA binding or decoding. An aminoacyl transfer RNA selected based on second messenger RNA codon in the A site enters and binds via codon-anticodon interaction with the help of EFTU GTP complex. This EFTU mediates the entry of aminoacyl transfer RNAs into A site. It binds as an aminoacyl transfer RNA EFTU GTP complex. First, EFTU binds GTP. This activated EFTU GTP complex binds to the transfer RNA, that is aminoacyl transfer RNA. So before the process, EFTU GTP are free. Then EFTU GTP binds and forms binary complex. Then it binds with aminoacyl transfer RNA and thus ternary complex is formed. This EFTU GTP then mediates this aminoacyl transfer RNA to the A site of the ribosome. So it binds with the ribosome as EFTU GTP aminoacyl transfer RNA complex. Binding of the aminoacyl transfer RNA to the A site is driven with the help of hydrolysis of GTP of the complex to GDP. At this point, EFTU is released, leaving the new transfer RNA in the A site. EFTS mediates the release of EFTU GDP from the ribosome and regeneration of EFTU GTP. So how the regeneration of EFTU GTP takes place? By EFTU EFTS exchange cycle. First, EFTS binds to EFTU and displaces the GDP. Then GTP binds to the EFTU and displaces EFTS. EFTU GTP is now ready to take part in another round of elongation. Now let us understand this EFTU EFTS exchange cycle with the help of this cycle. In the process of elongation, aminoacyl transfer RNA is mediated or escorted to the A site of ribosome with the help of EFTU GTP in the form of EFTU GTP aminoacyl transfer RNA complex. So once it enters the ribosome, hydrolysis of GTP takes place into GDP, an inorganic phosphate, and EFTU GDP is released, leaving aminoacyl transfer RNA to the A site. Then this GDP is displaced by the EFTS and forms EFTU EFTS. Then this EFTS is displaced by GTP and EFTU GTP is regenerated, and it is now ready to take part in another round of elongation. Again it binds with another aminoacyl transfer RNA depending upon the codon present in the A site, and the process continues. Now the second step of elongation, that is peptide bond formation. It is the second step in elongation. It is catalyzed by peptidyl transferase enzyme, an RNA based ribozyme that is integrated into 50S ribosomal subunit. In this reaction, the carboxyl end of the amino acid, or in further steps polypeptide chain bound to the transfer RNA in the P site, is uncoupled from the transfer RNA and becomes joined by a peptide bond to the amino group of amino acid linked to the transfer RNA in the A site, and energy for each peptide bond formation is derived from the high energy bond linking each amino acid to its transfer RNA. Now let us see peptide bond formation reaction. The amino group of the aminoacyl transfer RNA attacks the carbonyl group of the ester linkage of peptidyl transfer RNA to form tetrahedral intermediate. This intermediate collapses to form the peptide bond and release the deacylated transfer RNA. Peptide bond formation involves an acyl group O to N migration and converts an ester to an amide. So now let us understand the peptide bond formation reaction. Amino group of the aminoacyl transfer RNA, which is present at the A site, attacks the carbonyl group of the ester linkage of the peptidyl transfer RNA, which is present at the P site, and tetrahedral intermediate is formed. Then this intermediate collapses to form peptide bond. This is the peptide bond, and it releases the deacylated transfer RNA, that is deacylated transfer RNA is left at the P site. Peptide bond formation involves an acyl group O to N migration, and it converts ester bond into amide bond. And this reaction is catalyzed by peptidyl transferase enzyme. Now the third step, that is translocation. In the third step, a complex of elongation factor EFG, which is also known as translocase, and GTP, that is EFG GTP, binds to the ribosome, and after binding, three concerted movements occur, collectively called translocation. First, deacylated transfer RNA move from the P site to E site. Then second, the dipeptidyl transfer RNA in the A site move to the P site. And third, the ribosome moves along the messenger RNA, that is in the 5' to 3' direction by 3 nucleotide to place next codon in the A site. Now let us see some of the facts related to elongation. Elongation proceeds with single movement of the ribosome, each called translocation event. Movement of transfer RNA from A to P to E site is induced by conformational changes that advance the ribosome by 3 bases in 3' direction. After translocation events, the A site is empty and ready to receive the next aminoacyl transfer RNA. A site and E site cannot be occupied simultaneously. Thus, deacylated transfer RNA is released from the E site before the next aminoacyl transfer RNA binds to the A site to start a new round of elongation. The energy for each step along the ribosome is donated by elongation factor that hydrolyzes GTP. GTP energy is required both for the binding of a new aminoacyl transfer RNA to the A site and for its translocation to the P site after formation of peptide bond. Elongation continues, adding one amino acid to the C-terminal end of the growing polypeptide for each codon that is read, with peptidyl transfer RNA moving back and forth from the P site to A site as it grows. Now one may think, what will happen if there was no messenger RNA? If messenger RNA was not present in the elongation complex, the ribosome would bind to transfer RNAs non-specifically and randomly, and hence specific polypeptide would not be synthesized. Now let us understand this elongation process in detail. Aminoacyl transfer RNA that is selected on the basis of the second messenger RNA codon in the A site enters and binds via the codon-anticodon interaction with the help of EFTU-GTP complex. So this entry of aminoacyl transfer RNA is mediated by EFTU. Now a peptide bond is formed between this N-formyl methionine and the second amino acid. And this peptide bond formation is catalyzed by the enzyme peptidyl transferase. Then this N-formyl methionine is uncoupled from the transfer RNA at the P site and it joins to the amino group of the amino acid which is linked to the transfer RNA at the A site by peptide bond. Hence, deacylated transfer RNA is left at the P site and A site consists of peptidyl transfer RNA. Then the translocation process takes place and this is mediated by the EFG and GTP, which is also known as translocase. So in this process, deacylated transfer RNA now move from P site to E site. Dipeptidyl transfer RNA, which was present at the A site, moves to P site. The ribosome moves along the messenger RNA in the 5' to 3' direction by 3 nucleotides and places next codon in the A site. And technically, we can say that messenger RNA is shifted in this direction and exposes new codon to the A site. Now the A site is empty and it is ready to receive the next aminoacyl transfer RNA. This deacylated transfer RNA exits from the E site. A site is empty and it is ready to receive the next aminoacyl transfer RNA. A site and E site cannot be occupied simultaneously. Thus, deacylated transfer RNA is released from the E site before the next aminoacyl transfer RNA binds to the A site to start a new round of elongation. So, A site and E site cannot be occupied simultaneously. Hence, the deacylated transfer RNA is released from the E site before the next aminoacyl transfer RNA binds to the A site to start a new round of elongation, and this process continues in the cyclic manner till the entire messenger RNA is read or decoded. So, elongation continues, adding one amino acid to the C terminal end of the growing polypeptide for each codon that is read, with the peptidyl transfer RNA moving back and forth from P site to A site as it grows. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.